I'm Justina Studanlik, and it's my pleasure to be here today with Dr. Ralph Steinman, the Henry G. Kunkel Professor and Director of the Christopher Brown Center for Immunology and Immune Diseases at the Rockefeller University. Dr. Steinman has authored more than 400 scientific works, has been the keynote speaker at numerous symposia nationally and internationally, and he has received the highest accolades bestowed upon investigators in his field, including the Albert Lasker Award for Basic Medical Research and the Albany Medical Prize. Additionally, he is a member of the National Academies of Science and the Institute of Medicine. We are extremely thrilled that Dr. Steinman agreed to act as guest editor of Volume 235 of Immunological Reviews, covering dendritic cell subsets. He has assembled 21 outstanding reviews from leaders in the field. Today, we will be talking about dendritic cell research, beginning with how Dr. Steinman and his colleagues first identified this critical lineage of immune cells.
So first off, Justina, thank you and Gary Koretsky, very much for organizing this volume. I've always liked the approach that Immunology Reviews takes, which is to gather together a number of the experts in a field and have each of them outline their perspective of that field. So I'm very grateful to all 21 who wrote their chapters. So in terms of the discovery of dendritic cells, I think it was a situation very similar to most discoveries, which is that we didn't have a specific hypothesis. We were trying to tackle a problem. And for me, the problem had two aspects. One was during my medical training, I became interested in the importance of the immune system in disease. Infectious diseases, cancer, allergy, transplantation, autoimmunity, and probably more. And I wanted to understand how this system was controlled and how we could use immunology to design better treatments. And a related second challenge was that even with simple antigens, one didn't have a way of initiating the immune response. In other words, we knew infection triggered immunity, but as soon as you took individual proteins, it was difficult to do so. So my first approach to that problem was to pursue reports in the literature that intact antigens could be retained on the surfaces of macrophages. And the idea was that this would form a matrix that selected clones of T cells or B cells and activated them. This was before the days of MHC restriction and antigen processing. And I looked for, but failed to find, intact antigen being retained on macrophages. So at that point, I decided to move away from what was then the bread and butter of the laboratory, led by my colleague, my mentor, Zanville Cohn, which was the macrophage in the abdominal cavity. And I decided to look at spleen, an immune organ. And looking at what were called accessory populations in spleen, we spotted these very distinctive cells. And the first trait that we became aware of was that they were continually extending and retracting their processes, or dendrites, so we called them dendritic cells. That's how it began.
Great. So when your initial findings were published in 1973, what was the reaction of the immunology community?
So we thought the immunology community was going to be really excited because we thought this was a potential missing link in the immune system and could help explain accessory function, which we had not at that point demonstrated. But in fact, the broader community really wasn't interested that much, although a number of investigators all over the world began to study dendritic cells. In fact, if I could show you, this is one of the first reviews in 1980 that was in Immunology Reviews that Michel Nussensweig and I wrote. So there was some activity, but by and large, the broader community was interested in other fields, defining what T cells did, understanding MHC restriction and antigen presentation, for example. And in those studies, investigators commonly used T cells that were already activated or primed or immunized. And we were interested in a different problem, which is how do you initiate immunity? So I think, one, we were doing something different, and to the broader community were doing really important things, and that, I think, is the main reason why it took a while to gain recognition of these cells. And they weren't easy to work with.
Mm-hmm. So macrophages were widely held to be the primary antigen-presenting cells in the body until your work showed otherwise. Since then, researchers have understood more about the differing roles of macrophages and dendritic cells. So how have these ideas developed?
So I think some of the initial themes still pertain. So for example, with macrophages, their hallmark is that they are phagocytic cells, and they continually phagocytize at many stages of their life history. So that remains true to this day. What's expanding in macrophage biology is the number of products that they make, and also their different properties in different organs. Now, with respect to dendritic cells, still the initial theme, that is how you initiate immunity, is still a major one. But what's evolved, I think, is an expanding role for dendritic cells in controlling immunity, particularly controlling the type of immune response, the type of helper T cell differentiation pathway, and recently the importance of dendritic cells in tolerance, in silencing the immune system. So, yeah, the original themes are still pertinent, but we've added some new ones.
So you've had many successful investigators train in your laboratory over the years, and I was wondering if you could share with us, some information on a couple of them, and let us know if you've been surprised by any of the directions that the research has taken.
Okay, well, Justine, I'm glad you warned me you were going to ask that question, because that's a tough choice to make on the spot.
I know. not to select,
But I'd like to talk about two. Mm-hmm. Michelle Nussensweig, who was my first student to work on dendritic cells, and who's now professor of immunology, molecular immunology, here at Rockefeller. And I consider Michelle to be the leading investigator, particularly in molecular approaches to dendritic cells. And the second one I'd like to talk about is Gerald Schuler, who was one of the first postdocs to work on dendritic cell, and now runs the dermatology division at the University of Erlangen in Germany. Michelle was the first one to show that dendritic cells captured antigens and presented them. But what he has gone on to do in his own laboratory are some really surprising things. So first, he cloned DEC205, which was the first antigen receptor that dendritic cells were known to express in vivo. They've since been found to have a lot of candidate antigen receptors. And using that discovery, he obtained the evidence that dendritic cells in the steady state in lymphoid tissues were involved in tolerance. As I mentioned, the initial emphasis was all on immunization. And now Michel and his colleagues are doing fascinating work on the development of dendritic cells, figuring out for the first time where macrophage or monocyte and dendritic cell development diverges, which is in the bone marrow, according to their recent work. In Gerald's case, he too has done some really surprising things. In the lab, he discovered the phenomenon of dendritic cell maturation, which I think is one of the most overarching and important problems in immunology. So maturation means the differentiation that dendritic cells undergo when they encounter a particular environmental stimulus, and that enables them to initiate immunity and control its type, and also to control different forms of tolerance. So maturation is really important, and Gerald got that going with studies of Langerhans cells. Now Gerald is a premier investigator in using dendritic cells for immune therapy, including a system that he helped develop along with Antonio Lansavecchia and Federico Salusto. Gerald Schuler and another colleague, Nicky Romani, learned how to make dendritic cells from monocytes and then use them for doing cancer immunotherapy, which I feel is a really important area. Very demanding one, but Gerald's really leading the way there.
Excellent. So are there any areas that you think are understudied, or are there any technologies that would enable the field to move forward?
Oh, yeah, there's a lot.
Some key ones.
There are just so many labs that are doing interesting things. I think, while I've just mentioned it, the field of cancer immunology is one where there are relatively few investigators pursuing dendritic cells. In addition to Gerald Schuler, the group in Dallas, led by Jacques Bonchereau, are clearly very prominent in the field. But I think this is a particularly slow area of investigation. And it's not because of a lack of ideas. And it's not because it isn't a very logical avenue of research to understand how the immune system deals with cancer and to develop new strategies to deal with the disease. It's not because of a lack of ideas and people. It's simply a lack of support from the cancer community. And I think this is a real gap that needs to be addressed. Now, one of the issues that's addressed in this volume on dendritic cell subsets is the function of dendritic cells in different organs, and also macrophages, as I had mentioned. That comes out in the volume. There are articles about dendritic cells in skin, in lung, and intestine, even the eye. And I think this is really a good area to delve into in the future. Although just recently it's become apparent that the type of dendritic cell that's in these different organs is actually very similar to a type of dendritic cell found in the lymphoid organs, so that there is common ground in these different fields. Another place where I think we need new methods and there's a real gap is just on the tools that we use to study dendritic cells. So, for example, we're still missing monoclonal antibodies to some of the really important molecules on dendritic cells, not only for mice, but for studies in monkeys and humans. And then genetic approaches to dendritic cells, while they've begun, most notably by Stefan Young, who has a nice review on this in this volume, I think we need some new and better approaches to genetic approaches to study dendritic cells in vivo. And then finally, there's this theme of this volume, dendritic cell subsets. In other words, we know the subsets exist, but I don't think we really know what they do. And that's especially the case in humans, where most of the work's being done in mice.
So you have gathered 21 reviews for the volume, and I'm sure many more could have been included. And you've touched a little bit on some of the areas that are covered, but can you just give us a brief overview of the themes that are covered in the volume?
Yeah, there are a few recurring themes. One of them is dendritic cell development, which I alluded to just briefly. And that is, there's been a lot of progress in the last few years understanding how dendritic cells originate from precursors in the bone marrow and how their development diverges from monocytes. And specifically what's being found is that there are different hematopoietins for these processes, and there are progenitor populations that you can isolate with defined surface markers. So that comes up in a lot of the articles. Another thing that I mentioned that comes up in the articles is the function of dendritic cells in different tissues. And then a third theme is the different receptors that are on dendritic cells, particularly for antigen uptake and also for signaling the presence of microbes. And together these are the term pattern recognition receptors are often used. So if you really think about it, the markers that we use to distinguish the dendritic cell subsets often are receptors or lectins with a potential antigen uptake or a potential signaling activity. So that's a third recurring theme. One thing that doesn't recur enough is studies of dendritic cells in humans. There are some really lovely articles, particularly by Jacques Bonchereau. The topic is covered also by Marc Delot and some others, but it's still underrepresented. And we really need to bring dendritic cell science and immunology science in general into the patient more effectively than has been done today.
Great. Well, thank you very much. It's been a pleasure speaking with you, and to our listeners, I hope they'll join us again.
Thank you, Justina.
Thanks.
God bless.